Yeah, my name is John Wall. I'm Director of Operations at VRL, and I'm going to be talking today about rodent environmental health monitoring, and specifically some studies we're doing to evaluate and identify an optimal Sentinel swab media for Sentinel-free soil bedding testing. Okay, so the agenda is I'm going to give you a brief overview of what we do at VRL for people who don't know about our company, and I'll give you a couple slides of background about the use of FSSB testing in rodent health monitoring, how it's testing has evolved to having more and more of that. We're receiving more and more samples. I'll give you a little background. And then I'll show you some intermediate data from a contact media experiment where we were testing 3 contact media that had previously shown good performance in a previous study. And then I'll give you some conclusions and next steps. All right, so VRL is an animal health diagnostics company. It's been in business for about 30 years, and we're focused on laboratory animals. We have 2 facilities in the United States, one in San Antonio, which focuses on non-human primate testing, which has a large percentage of the market for an NHP business. And at VRL Maryland, where I'm working, we focus on rodents and other small animal models. So we also do animal health monitoring for zebrafish, rabbits, gerbils, xenopus, and any other small animals that you would like to have tested. We work with CROs, pharmaceutical companies, biotech companies, government agencies, university breeders, a whole range of people in the industry. And we have 2 main testing capabilities: animal health monitoring, which is maybe more than 50% of our business, and preclinical bioanalysis testing, which is less than 50% of our business. And this slide shows essentially the range of testing that we do. For animal health monitoring, we have a large array of PCR and serology-based testing that we can do. We also provide necropsy and histopathology services, bacteriology and parasitology, the whole range of kind of testing that you would need for a reasonable animal health monitoring program. We also provide, as I mentioned, preclinical bioanalysis. This includes clinical chemistry, hematology, coagulation, panel studies, urinalysis, cell line pathogen screening, sterility testing, cell line authentication. Okay. And this slide just shows just gives you an idea of we have a strong commitment to various aspects of this business. We have a strong commitment to superior customer service. We spend a lot of time working one-on-one with our customers to help them develop their monitoring programs. We deliver reliable results. All of our positive testing is always confirmed by orthogonal methods before we report them to you. We also have a convenient data access system, a client portal, online testing results, online sample submission that are easy to use, and will help you with getting your testing done efficiently. And maybe kind of an emphasis on fast turnaround times. We think we have the best and the fastest turnaround times in the business. So for PCR testing, for instance, we can get results within 48 to 72 hours. If it's an emergency situation or a quarantine situation, within 48 hours. Microbiology within 3 to 5 days, clinical chemistry within 24 hours, and we can accept samples on weekends. I also forgot to mention that for this, we do provide, we work with customers if they have PCR tests that we don't have in our catalog. We can work to develop those methodologies for you. Okay, so on to my presentation. So Sentinel-free soil bedding testing is something that's becoming more and more prominent in the industry, and it's replacing soil bed sentinels. And if you're not familiar with the service, this slide gives you a little brief description. Basically, you have a periodic transfer of soil bedding from multiple animal cages into an empty sentinel cage. That sentinel cage contains swab or filter material.
Maybe I think it's hard for us to hear in the back. Can you just get the...
Oh, closer? Oh, I'm not sure you all want that, but okay. Okay, so where was I? So the sentinel cage will contain swab or filter media to absorb infectious agents, the genetic material, and then after a predetermined interval, often 3 months or a quarter, so the swab or the filter is collected and tested for the presence of infectious agents. And so the mechanism of action is essentially the filter or swab will absorb fecal, oral, or urine matter, and that will adhere to the media, and then it's extracted out later. And all the testing is done by PCR. which obviously gives you a full range of infectious agent testing, virus, bacteria, and a whole range of parasites. So there are several advantages to SFSB testing with respect to sentinel testing or exhaust dust testing. One is it's compliant with the 3 R's initiative. There's no live animal sentinels that need to be used. It can be used in any type of rack. You don't need a dedicated rack that is associated with EDT. It's inexpensive compared to the other testings. And recent data, especially, I'll show you a couple of examples from JLAS articles, show that this kind of testing is more sensitive than sentinel testing and environmental dust testing. For one, it doesn't rely on aerosolization of infectious agents, as you would see with EDT, and the sensitivity doesn't depend on animal age or strain or shedding patterns in comparison to the sentinel testing. There are certain, I wouldn't even call them disadvantages necessarily, but concerns. So it's important to develop a uniform technique, which may not exist yet, where you would make uniform the amount of bedding that would be used, frequency of transfer, frequency of the bedding disposal, the stirring or shaking methodology is probably variable, in addition to the collection media. So I will kind of identify in a slide exactly how we did it, but our testing is focused on that collection media, finding the best collection media. So this is a represented data from recent literature. These are both JLAS articles, and which essentially supports the use of SFSB testing in comparison to sentinel testing. This one from JLAS Win et al. In 2022 has several studies. This is just one example study where in this column you have direct sentinel mice testing. This column filter PCR testing, where you can see a much higher percentage of detection was gotten in comparison to direct animal testing. So that clearly seems to be advantageous compared to the sentinel. They did note that that opportunists and infectious agents that are at low level are still not necessarily detected very efficiently, and that's of course a reason for us to try to optimize that. And then there was another article from O'Connell et al. Where they tested 2 filter media in comparison with sentinel mice. In essentially all cases, the sentinel testing was the sentinel-free testing was superior. They did not necessarily identify which filters were used. In some cases, I think that they were proprietary filters, so that's part of the rationale for why we're doing the testing that we're doing. So the testing parameters of interest. We wanted to find out the best contact media for pathogen collection, how long an exposure is needed for reliable detection, and how does swab testing compare to exhaust dust testing, at least in our experiment. So this is the experimental setup. So we actually tried to, in previous studies, we basically collected pet shop mice from a single source and done testing. And often we had cases where there was not a wide array of infectious agents that we could detect. So in this experiment, we actually collected from 4 separate locations, in fact, from 4 separate states, to try to get a wider array. And embedded in this also is actually just a higher N value. So we have 4 separate independent tests that are performed together. So for each of these sources, we have its own contact sentinel cage. And then we also collected a combined cage that represents the whole combined sentinel for the entire rack. We used the Technoplast Easy Flow system. It was cellulose bedding. We did time points 1 and 2 weeks and 1, 2, and 3 months. Of course, I've only had the 1 month data now. And we also compared the system filter testing at 1 month with our SFSB testing. And this is the overall sentinel cage protocol. I'm not sure how easy that is to see. So essentially, we did day 0. We did live animal testing from feces and fur swabs, etc. And on day 3, we created our sentinel cage media. We added 12 of each type of media that we were going to be testing to the bottom of the cage. We added essentially about 100 milliliters, 6 tablespoons of soil bedding to each of those sentinel cages and mixed it up, and that started the experiment. And so for the PCR testing time points, this is the protocol. Essentially, you'd shake the sentinel cage 10 to 15 seconds. You remove the contact media, 2 of each type, 1 for DNA, 1 for RNA. And then we would dispose of the sentinel cage bedding, leaving the remaining media, and then add additional soil bedding to the sentinel cages, and then change the animal bedding. And then we did do cage changes weekly. So when we weren't doing PCR testing, the protocol was essentially transferring additional bedding from the animal cages to the sentinel cage, shaking the sentinel cage to make improve contact with our filters, and then change the animal bedding. So a note about the contact media selection. This was not the first experiment we did with contact media. We'd done some previous experiments before where we tested two types of swabs, a flock swab and a polyester swab, and we tested several filter types, cellulose-related ones, several cellulose-related testing types, synthetic cotton and polyester and nylon. And then we did more limited study from one source. And I mentioned we didn't get that many of that much of a wide array of infectious agents, so we had limited results. But we did get clear kind of patterns. Clearly, the polyester swab was more sensitive than the flock swab, and the cellulose-based filters were more sensitive than the others. So that's what drove the current study that I'll be describing. So this data shows the initial PCR results from the animals, and you can see there is a wide array of infectious agents, several bacteria, four prominent virus, and several parasites. Disappointingly, we did not get differences between these different sources, and we later found out that even though we looked in 4 different states, I think they came from only 2 separate breeding facilities, so we didn't get that wide array. That doesn't really affect the experiment. They still represent a wide array of infectious agents and 4 separate independent tests. And this is a result, this is a representative result. This is from source B for what happens at 1 to 2 weeks. And we hadn't really tested this before, but surprisingly, you do get really good uptake and ability to detect infectious agents even up to in as little time as 2 weeks. So especially with media 3, in this case, it was 9 out of 9, 100% detection. The swab media 2 was slightly worse, and the swab was slightly worse in this case. But this is just representative data. This is the complete data set for early detection, and I'm going to focus on the 2-week time point. So in this case, did kind of hold true that media 3 had very good detection, 94% detection in comparison to the direct animal testing, whereas the other 2 were slightly worse. And this t-test comparison is not really with respect to percent success rate, but really with respect to copy count, because all these were PCR results where we also did a standard curve and generated apparent copy counts. So it's not really clear if this has any kind of utility for animal health monitoring, but we thought it was an interesting result, and it's also a good measure, we think, to compare relative sensitivities of the media. So from the 2-week data, all 3 media seem to support some level of detection of the entire range of infectious agents, and media 3 was slightly better. And the next 3 slides are essentially going to show you the 1-month time point for bacteria, virus, and parasites. It's a similar story for each of these 3, but so for bacteria, media 3 again was the best at showing a success rate. So at 1 month, in 95% of the cases, we were able to detect what was originally detected at time 0 from the animals. And the swab, polyester swab, which we often get from customers, actually was also fairly efficient. You can see the copy count data where there's these blue and green, which represent swab and media 3, were clearly superior in all cases to media 2. This is also reflected in the virus detections, where media 3 and the swab were detected at 100% rate. Media 2, again, was lagging. And with parasite, the same story. So media 3, 91%. Always media 3 was either as good as or better than the swab. And this is also reflected in the graph there. So this is the summary of the 1-month detection data from all 4 sources. and media 3 and the swab were the better ones, and they also kind of supported higher PCR copy counts, and this was statistically significant with the t-test. As a side note to our study, we had done this, as I mentioned, in the Techniplast Easy Flow filter system, and we compared our results with the Techniplast filter exhaust dust testing approach. And at least at the 1 month time point, any of these media and the SFSB approach was superior to the exhaust dust testing approach by a pretty dramatic margin, I would say. And so, for instance, the Techniplast was only able to detect the infectious agent at 36% rates, whereas the others were, 80 to 90%. All right, so conclusion. So both the polyester swab and the cellulose-based filter have high sensitivity for pathogen detection. Media 3 is slightly better. Effective detection was observed as early as 1 to 2 weeks post-introduction, and we had a 90 to 100% detection of bacteria, virus, and parasites at the 1 month time point. Of course, we're very much interested in seeing what happens at 2 and 3 months, especially the 3 months, because that's sort of more in line with what samples we receive. We usually receive quarterly testing and quarterly sentinel media. So we need to do. We think we need to do just a little bit more further testing to finish this evaluation of the best contact media. We'll be finishing the study shortly, and we may do one more follow-up study where we also look into some of these other factors, essentially the manipulation factors. But we think that based on this study, we will be able to provide our clients with the best contact media to support this type of testing. So thank you for your attention. Definitely willing to answer your questions here or at our booth, which I believe is 2105. Thank you. Yes.
Would you use that with static cages as well, or just IVC cages?
The question is whether it can be used with isolated cages as in addition to the IVC cages. Yeah, that's what we think is one of the major advantages of this kinds of testing is you don't need that. It can be used with any kind of cage setup. Sure. Were all of the questions answered? Oh, well, there's another one. Okay. I'm not sure I understood. So are you asking whether or not one sample is sufficient to do all the testing? Yeah, I guess it would depend on your. The question is, can you send out everything together in one tube, all the contact media, or not? I think it really depends on the point of your testing. I mean, if you need to have separate infectious agent detection or a health monitoring result, they need to be separate, or they're separate studies or separate purposes. You need to keep them separate. Otherwise, I think you can combine them. Yeah. All right, thanks.